Imagine you're writing a book, a really long book. Let's say War and Peace, and halfway through, you realize you spelled the main character's name wrong. In the old days of genetic engineering, like we talked about in the last episode, fixing that mistake would be like trying to rewrite the page with a shotgun. You'd blast a hole in the book and hope the new word landed in the right spot. It was messy, it was expensive, and it took forever. But what if you had to find and replace? What if you could just type in the error, hit search, and the computer would find the exact sequence of letters among millions of words, delete it, and type in the correct one? Well, around 2012, we found that tool inside a bacteria. It's called CRISPR. It's cheap, it's precise, and it's the most powerful technology humans have discovered since the splitting of the atom. Today, we're going to learn how to edit the source code of life. So where did this sci-fi technology come from? Did we invent it in a high-tech lab? No. We stole it from bacteria. See, bacteria get sick just like we do. Their enemies are viruses called bacteriophages. These viruses land on the bacteria and inject their own DNA to hijack the bacteria's factory. It's a hostile takeover. For billions of years, bacteria have been fighting this war, and they developed a secret weapon, an immune system. When a bacteria survives a virus attack, it keeps a tiny piece of the attacker's DNA. It files it away in a special archive inside its own genome. This archive is called CRISPR. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. Don't try to memorize that, just call it CRISPR. Think of CRISPR as a wanted poster, or a mugshot. Now the bacteria has a partner in crime, a protein called Cas9. Cas9 is the executioner. It holds the mugshot, the RNA, and patrols the cell. If it sees any DNA that matches the mugshot, it cuts it up. Virus destroyed. This was just a tool, a cool biological fact, until two scientists, Jennifer Doudna and Emmanuel Charpentier, asked a $1 trillion question. What if we gave Cas9 a fake mugshot? They realized that Cas9 is programmable. It doesn't care what it cuts. It just cuts whatever matches the mugshot you give it. So, if we want to cut out a gene that causes cystic fibrosis in a human, we just manufacture a piece of RNA that matches that bad gene. We give it to Cas9 and we inject it into the cell. Cas9 scans the billions of letters of your DNA, finds the exact spot that matches, and cuts it. Once the cut is made, the cell panics. Oh no, my DNA is broken, and it rushes to repair it. And this is where we trick it. We can provide a new piece of DNA, a template, and the cell will use that to patch the hole. Boom! You didn't just delete a gene, you rewrote it. This is why CRISPR is a revolution. The old ways were like using a hammer. CRISPR is like using a cursor. Now you might be thinking, okay, cool, but I don't have $1 billion lab. Here's the craziest part. You don't need one. Before CRISPR, genetic engineering cost thousands of dollars and took months. With CRISPR, you can order the materials online for about $75. It is so cheap and so easy that high school students use it for science fair projects. There's a whole community of biohackers working in their garages trying to engineer glowing beer or color-changing flowers. It is democratized science. It means the power to change life isn't just in the hands of megacorporations anymore. But that's also the scary part, because if it's easy to cure a disease, it might also be easy to make something dangerous. CRISPR won the Nobel Prize for Chemistry in 2020. It's the defining technology of our generation. We have the scissors, we have the search function, we have the code. So what do we do with it? We could use it to design tastier apples. We could use it to make algae that eats plastic. But the biggest target, the most controversial target? Us. What happens when we turn this tool on ourselves? Can we delete cancer? Delete HIV? And what happens if we try to delete shortness or bad eyesight? In the next episode, we're going to look at the people who are alive today because of this technology. We're going to talk about the cure. I'm your host, Novak. This is the GeneLab. And remember, your DNA is no longer written in stone. It's written in pencil. Stay curious.